So next part for you to consider is how you're going to do your in vitro testing of your material that you've manufactured. And so we touched on in the lectures what your options are for in vitro modeling, so lab-based modeling. And so what I want you to see you do here is to pick the appropriate cell types and talk about how those cell types should behave on your material if they're doing the right thing. And then what I want to showcase is what we touched on in the complexity part of the lecture most recently, but we didn't get to go through in a lot of detail, is when we test vascular constructs in the lab, we're using this kind of bioreactor system that seeks to mimic more than one aspect of the biology, so not just cells stuck to a plastic plate. So our reactor allows you to put a three-dimensional graft in the reactor. And then what we're trying to do is put kind of pressure and flow that are replicative of what you find in a human. And then we've got sensors attached to them so that we can see that output, and we've got kind of concrete measures that the flow and the pressures we're putting in are appropriate. And then we can seed cells in via the cell seeding port. So you've got your graft, you can attach it into your graft chamber, connect it to the pumps, so you've got a closed system, and then after that you can do cell injections through the cell injection port. The cells will flow around the system and adhere to the grafts, and we've got the opportunity to seed kind of multiple cell types. And as I touched on in the lecture, for vascular applications we're looking at both endothelial cells and smooth muscle cells. We want the endothelial cells in particular to be on the top of the graft in contact with what would be the blood, and we want the smooth muscle cells ideally in the wall of the graft or inside the graft. So the framing of it then is, okay, we've got a 3D scaffold, we've electrospinal silk conduit. Ideally we want to seed kind of smooth muscle cells in the wall of that graft, so it has to be porous to allow for that. And then we want to put a layer of endothelial cells on top of that to best replicate what we're finding in tissue. So our bioreactor, as you can see, has some pulsatile flow to it, and so in the kind of supplementary video you can see it pulsing along with that flow. And then the outputs of the graft are, well, the good thing is we can culture it like that for several weeks, so 2-3 weeks is within the scope of what we've done so far. And then we can look inside the grafts, work with microscopy or histology, and see how the cells behave. So I touched on this picture in the lectures, but you can really clearly see smooth muscle cells in the wall of the graft, just as but the biology suggests. And then on top of that, the really clear endothelial cell layer. So a much more kind of advanced way of doing it than just tissue culture plastic. That allows us to study like crosstalk between those two cells. It allows us to study response to injury. So one of the things we're interested in is injuring that endothelial layer and then kind of seeing it recover. So asking some specific biological questions around how it behaves in an in vitro model. So and particularly relevant to this material, it allows us to measure how fast it will re-endothelialize, because that's really the main outcome that's linked back to its performance if I go and implant it in an animal model or in a human. And I want your thinking about your assignment to reflect that as well. Like, it's one thing to have a model, but what is the outcome from that model that best ties us back to what would be the performance down the line? So for us, it would be speed of re-endothelialization. So maybe we'd seed endothelial cells, we'd watch them adhere, and then we'd watch them grow over time. So the final thing to say about the model is that this flow and pressure that we're applying to it does really significantly change the cell behavior. So you can see in these ones where there's either no flow in this middle part where it's static, or mid flow or high flow, you get alignment of the endothelial cells that is more replicative of what you find in real tissue. And then the endothelial cells start expressing cell surface markers that they would normally express in tissue, so like ICAM1. And so this gives us the sense that under these flows and under these pressures, they are more like we would find in real vasculature. And so then if our material is doing better in those conditions that better approximate that tissue in these more complicated conditions, then we have greater confidence that the material is doing well and that we can carry it forward. Kind of justifies going to the next phase, which might be an animal model. So to recap, I want you to think about what is a good in vitro model. Select a good representative cell type that is appropriate for your tissue selection, and then talk to me about what would be the outcomes from those cell models that show that your material is doing well and that are linked to being able to take it to the next phase.